digestive system. So first of all, the human alimentary canal runs right through from the mouth to the anus and includes everything in between. And today we'll be focusing on the mouth, including the salivary glands, the epiglottis, the esophagus, stomach, the small intestine, and the large intestine, and then the rectum and the anus. So first of all, I'm going to look at intra and extracellular digestion. So how do we get nutrients from the food that we eat? So first there's intracellular digestion. This happens within the cell. An example is bacteria being engulfed by the phagocyte, taking the bacterium in, breaking it down. Cells that line the gastrovascular cavity engulf it, and digestion takes place inside these cells. Lysosomes are organelles that are involved in the digestive process. The breakdown is basically the breakdown of complex food to simple form inside the cellular cytoplasm. And then extracellular digestion is the form of digestion that happens within the digestive tract outside the cell. So during digestion, we break down food into smaller parts called nutrients that our cells can then utilise. Two other forms of digestion can be categorised as mechanical and chemical. So mechanical is referring to the physical breakdown of food into smaller pieces, such as chewing, emulsification, and churning of the stomach. And chemical referring to the enzymes, which are biological catalases that are released to break down food particles, increasing their surface area for easier digestion and aiding the absorption of nutrients. The catalases speed up the rate of digestion by facilitating and bringing down the activation energy. The most important part of the enzyme is the active site, which is specific to the shape of the substrate that it will receive. The enzyme will have a complementary shape to the active site, which we will look at next. So first of all, the hypothesis that was first introduced was the lock and key hypothesis, and this is basically where it says the enzymes are specific. Not every enzyme can facilitate the reaction of every other reaction. The substrate and enzyme site have to have complementary shapes, as this diagram shows. The fit has to be very close. Although since then another hypothesis has been introduced, which is induced fit, which means that there may be one enzyme able to facilitate reactions of a simple shaped substrate and is able to slightly alter its shape in order to fit, as shown in that diagram. The hypothesis is much more accepted as it replicates what happens naturally in nature. So now we're going to look step by step in more detail of the different functions of each part of the human alimentary canal that I mentioned at the beginning. So all food starts off in the mouth. It's the first place in which the process of digestion occurs. The physical breakdown or mechanical act is the chewing of the food by using the jaw, tongue and teeth to break large parts of food down into smaller, much easily digestible particles. The salivary glands excrete saliva, which is the chemical digestive function of the mouth, which also it has an enzyme called salivary amylase, which acts upon carbohydrates almost straight away. Saliva also contains mucin, which makes the mouth and throat slippery in order to soften the lining of the throat and lubricates the food. Mucin also has like a buffering capacity, which neutralises acids to prevent the teeth from decaying. When the food has been chewed, it's moulded into a lump called a bolus, ready to be swallowed. So next we come to the epiglottis, which is a small flap which basically just controls swallowing, prevents choking. That's its main function. After that we have the esophagus, which is a muscular tube around 20 to 30 centimetres long, which joins the mouth and stomach. It has two layers of muscles, both smooth and longitudinal, and skeletal muscles. Food is then moved down by peristalsis, so that's a series of involuntary wave-like muscle contractions which move food along the digestive tract, and this is so strong that it actually enables you to drink upside down. After the food has passed through the esophagus, it reaches the stomach. The stomach can be described as a muscular bag which can hold around 1.5 to 2.5 litres. Food is temporarily stored here, and both mechanical and chemical digestion takes place here. So the mechanical would be when the stomach churns, where partially digested food is broken down into nutrients ready for absorption, and the chemical element is primarily due to hydrochloric acid, which has a pH of 1.5 to 3.5, so quite acidic, which helps to break down food. Gastric secretions such as pepsin, hydrochloric acid, mucus are also found here. So the main digesting enzyme pepsin is activated upon the exposure to the hydrochloric acid, and as mentioned before, enzymes are specific and each one can facilitate the digestion of certain food types. So amylase will digest carbohydrates, breaking them down into sugars, and is produced both from the salivary glands and the nearby pancreas. Maltase is the enzyme that breaks down maltose into sugar, which the body needs for energy. Lactase is the enzyme which breaks down lactose and is produced by cells called enterocytes, which are found lining the intestinal tract. Lipase is the enzyme that breaks down fatty acids, fats into fatty acids and glycerol, which are produced in the salivary glands and the stomach. And protease, which the name would suggest, breaks down proteins into amino acids as well as digestion. This enzyme also helps the body with cell division, blood clotting and immune function. So I'm just going to look at the structure of the stomach wall. So it has 4 layers: the serosa, a collagen layer protecting the stomach, muscle layers, submucosa and mucosa. So the mucosa is the inner layer, the stomach lining, and it's bumpy and not smooth. It also has rugae, which increases surface area to produce more gastric secretions to digest food chemically by releasing hydrochloric acid and pepsin. And it also contains glands which help to digest food. It's made of epithelial cells that produce secretions. The next layer is the lamina propria, which is made of loose connective tissue such as collagen and elastin that supports the delicate mucosal epithelium, allowing the epithelium to move freely and provide immune defence. It also acts as a barrier to stop unwanted materials and organisms from getting food to the body. It also lies between the submucosa and the mucosa. The mucosa surface layer is covered by a single layer of columnar epithelial, and it contains gastric glands and pits, which are made of peritial cells that secrete hydrochloric acid and secrete pepsinogen, which is an inactive form of pepsin. A thin layer of mucus protects the stomach from digesting itself with pepsin, preventing self-digestion. The stomach wall contains gastric glands which produce mucus, aiding the stomach wall from the secreted gastric acids. So the next part, the submucosa, that's a thick layer of loose connective tissue in 3 layers, oblique, circular and longitudinal, that surrounds the mucosa. It contains blood vessels and nerve tissue endings which signal the brain when the stomach is full. And then the serosa, which is the outer layer of the stomach, is made of a simple layer of simple squamous epithelium and a thin layer of connective tissue. It has intertwined collagen fibres that hold it together and blood vessels that line it. And its main function is to protect the organ from any friction as it expands with food and moves, so when it's churning, when it's mixing and propelling food. So chief cells will secrete pepsin to digest proteins found in the gastric glands. Peritial cells we've looked at already, and chyme would refer to the semi-liquid, half-ingested food that's been mixed with these gastric juices, ready to be released by the sphincter in the small intestine, which is the first part of the small intestine. So here's the small intestine. So it's split into 3 sections. Most of the chemical digestion takes place here. So the first part, it would be the duodenum, which is like a C-shaped structure, which is the first part. the jejunum, which is the middle part, and the ileum, the last and longest part of the small intestine. It's not until the big macromolecules have been broken down that the molecules can then be absorbed into the blood to the small intestine. And in order for absorption to be more efficient, a very large surface area is required, which the small intestine has, with it being very long. It also has folds called plicae made of villi, which are made of epithelial tissue, or microvilli, which also increase the surface area. So first of all, the duodenum, that's the first place that food comes after it's been mixed with stomach acid, where it comes here to be mixed with bile, which is made in the gallbladder, and digestive juices from the pancreas. The middle section, the jejunum, the main job is to absorb sugars, amino acids and fatty acids. There are blood capillaries also needed to be present, and what they do is they take away what's been absorbed. The capillaries take glucose and amino acids to the blood. Fatty acids and glycerol would prefer to go into the lymphatic system, and the equivalent of capillaries in the lymphatic system are lacteals, before returning to the bloodstream, distributing them to every cell in the body. So as mentioned before, the structure is very long, it's around 5 metres. Both the duodenum and jejunum have got circular folds which increase the contact surface with the food, and the vast majority of the small intestine is covered in villi, which are about 1 millimetre long, that increase the surface area of the tissue available to absorb nutrients from the gut contents. They also contain blood capillaries. So each villus absorbs and transfers nutrients to these blood capillaries. Some nutrients like iron require a transport protein called like transferrin, and fats need lipoproteins. And these are then transported through the lacteal of the villus. The role of the lacteals and capillaries in absorption in the ileum is that that's got small villi again that line the walls of the intestine. They absorb nutrients into capillaries of the circulatory system and the lacteals of the lymphatic system. And if you were to look closely at villi, they've got little capillary beds as well as lymphatic vessels called lacteals. Fatty acids absorbed from broken down chyme pass into the lacteals, and then other absorbed nutrients enter the bloodstream through the capillary beds, and then they're taken directly to the liver via the hepatic vein for processing. There's not really much else to say about the last part. It's the longest part, the ileum, of the small intestine, and unlike the duodenum and jejunum, it does lack the circular folds. It contains lacteals and capillaries, as mentioned before, to aid in absorption, and small villi. Another important job for the ileum is to absorb electrolytes from calcium, which helps to build hair, teeth and bones, B12, things like that. That's production, and the production of red blood cells and the remaining bile acid that's transported back to the liver through the bloodstream. The next part is the large intestine, also known as the colon. So it's much thicker than the small intestine. The name large refers mainly to its thickness rather than its length. Because the small intestine has already at this point absorbed most of the digestive substances, the large intestine will lack villi because it doesn't need any. It does, however, have billions of bacteria inside, which are vital for the immune system in killing harmful pathogens. They also produce vitamins and turn fibre into energy. The main difference in the large intestine that is it also has many goblet cells which secrete mucus just to aid in the digestion. So chyme is transported from the colon by, again, peristalsis, from the ascending colon, which is seen on the left of the diagram, to the transverse colon, and then to the descending colon, where on its way, water and salt are eliminated from the chyme and mucus is added to it to help with the excretion of waste. And then the end result at the end of the rectum are all the substances left that can't be absorbed any further by the body. They're held there and then they're secreted through the anus. So the general layers of the large intestine is similar to the stomach. So they have mucosa, which have I've got pits which secrete mucus called intestinal glands, and they're consisted of 3 sublayers. Simple columnar epithelial, which is where the goblet cells are found, which produce mucus. Then the lamina propria, which are deep to intestinal glands, and again submucosa. The next layer would be the muscularis externi, which has both circular and longitudinal layers, and the deepest layer is the serosa, made of simple squamous epithelium. And finally, an organ that does assist with the digestive system is the liver, that I've spoken about a little bit before. So the liver processes nutrients absorbed from the small intestine via the hepatic nerve, as mentioned before. Bile from the liver that is secreted into the small intestine has the job of digesting fats and some vitamins, and it also metabolizes fats, proteins and carbohydrates. And that's it.